This is interphase, and most of a cell's life is spent in interphase. In G1, the cell grows by producing more proteins and organelles. In S, DNA replication occurs. In G2, the cell prepares for cell division with the appearance of centrosomes. And in M, mitosis and cytokinesis occurs. There are 3 checkpoints in interphase, which is G1, G2, and M.
In this cell, we observe 4 haploids, which will duplicate and become diploids that have a ploidy state of 2 n = 4. This tells us there are 2 types of each diploid, and there are a total of 4 chromosomes.
In prophase, the nucleolus disappears and chromatin compresses into chromosomes, which are visible in this phase. The separation of centrosomes takes place, as well as the formation of mitotic spindles. At the end of prophase, the nuclear envelope disappears. In pro-metaphase, the nuclear envelope breaks down and the nucleolus disappears. In metaphase, there is no nuclear envelope, and there is thick coiled chromosomes, each with 2 chromatins that are lined up on the metaphase plate. The centrioles are at the opposite ends of the cell, which allow the spindle fibers to run from centriole to kinetic cores.
Anaphase is the shortest phase of mitosis, and in this phase, the sister chromatids separate as microtubules pull them apart, causing the microtubules to shorten.
In telophase, the chromosomes are in a cluster at opposite ends of the cell. The nuclear envelope reforms in order to create 2 new cells. In this phase, the supercoiled chromosomes unravel and become visible again. In other words, chromosomes unfold back into chromatin. Cytokinesis is the process in which the cell divides into 2. With the nuclei being at opposite ends of the cell, the cytoplasm divides and the cell pinches in the middle, which leads to a cleavage furrow. The cell is pinched until it separates for 2 daughter cells to be created. Without cytokinesis, the growth and development of new cells is impractical.